So welcome to the International Chromosomal Instability Seminar Series. Our speaker today is Professor Sarah McClelland from the Barts Cancer Institute. I'm now going to hand over to Sarah.
Wow, that was so brief, Amy. Thank you very much. That you caught me slightly off guard. But here I am. Thanks for the intro, and a big thanks to Amy actually for stepping in last minute here to introduce this series on behalf of Florian Markowitz and others who all couldn't quite make it for various different reasons. So yeah, it's great to see all the names joining. I can't see everyone's pictures particularly with the screen setup I've got, but some familiar names and a lot of new names. So hello everyone. As you can see, I've put this post here up. So I'm really honoured to be part of this really exciting lecture series on chromosomal instability. We've had some great talks already. We've got some great ones coming up. As you can see here, I think unfortunately the first one here in January can't happen as advertised at the moment, but hopefully the SIN network will be in touch with you with further details of upcoming lectures. So yeah, once again, thanks very much for being invited. Hope you find this interesting. I think you can write questions in the chat and then Amy will read those out to me at the end. As I can't see that, or I think you can also probably just put your hand up and ask a question in person if you prefer. Will we be able to do that, Amy? We can keep an eye on we can keep an eye. So if you prefer to ask like that, please do ask me some questions. Great. Okay. Let's see if I can get my cursor to the correct screen and start my talk. No. I've lost my cursor, so I can't actually find. Oh, there it is. Okay. I can't actually. Oh, I'm on Zoom. There we go. Okay, I was stuck on Zoom. Okay, right. Well, so our lab works on sort of various facets of chromosomal instability, with a particular interest in understanding the mechanisms that drive chromosomal instability, or SIN, as I will call it most frequently, in cancer across multiple cancer types. And yeah, so I'm at the Barts Cancer Institute, as Amy said. That's part of one of the many universities in London. This one is Queen Mary University of London. And we're also part of the CRUK City of London Cancer Centre in collaboration with several other universities and institutes. And I thought I'd like to start my talk just introducing where on earth this actually is. So we're right in central London, actually, is where the medical campus is, which is where we're part of. Despite all my practicing earlier, I can't see my cursor now, so apologies for that. You'll just have to be directed on the slides by my voice. So, but what you can see is sort of some very nice images of very old brick buildings and other buildings. The lower one on the left is the Barts and London Hospital, which is one of the oldest hospitals, over 900 years old, and the medical school is associated with that. And the map on the right, we're right in the heart of London, in central London, surrounded by other fantastic research institutes, Francis Crick Institute, UCL Cancer, ICR, and it's a really nice, fantastic place to work in terms of science, because we've got a lot of people right on our doorstep, a lot of collaborations and a lot of resources here. So that's where we are, to put us in context. Right. So one of the main reasons we and many others have been interested in understanding what drives chromosomal instability in cancer is because it's pervasive and extensive across nearly all cancer types. There's many ways you can visualize this process and the results of this process, which is alterations in our genomes, in our chromosomes. So the two movies on the top left are ovarian cancer cell lines, which we've tagged the DNA, their chromosomes, with fluorescent histone, so we can track them as they make their way through mitosis. And what you can see in both these particular examples, they're two different cell lines. They're making obvious mistakes during mitosis. That is, if you've spent enough time looking at normal mitosis, they shouldn't have these strands of DNA chromosomes being pulled apart at the final stage of anaphase here. The other one on the top right has trouble aligning its final chromosome there. Eventually, when it does, it goes through with two anaphase bridges. So this is pervasive. We see this in ovarian cancer cell lines, at least about 1 in every 2 divisions. The cells will make at least one of these large visible errors that we can see with microscopy. So this, the consequences of this is also seen in the chromosomal alterations that are also well known to be associated with cancer. And on the right top left, we have a normal human cell's complement of chromosomes, 2 identical copies of each of the maternal and paternal alleles. However, in the progression towards cancer, and this is an example of a high-grade serous ovarian cancer again, there are a myriad of alterations in the given cell, which include structural alterations or numerical alterations, if we want to define them like that at the moment. Which you can see, this is a multicolour fish where we can actually label each parts of each chromosome with a differentially coloured set of fluorescent probes, and we can actually quantify alterations at a single cell and single chromosomal allele level like this. So what that actually allows us to do then is not just look at one cell here, but we can start to appreciate the heterogeneity that this process, since it's an ongoing process, can generate. And that was really visually revealed to us when we first did this MFISH on this ovarian cancer cell line. So you can tell I'm in London, so hopefully you can see hear the police car zooming past outside my window right now. Hopefully that doesn't come through too much. It's a bit off-putting to me. So what we can notice is of each of these are 3 cells picked randomly from the population, and we did about 25, and there's a very obvious heterogeneity at cell-to-cell level in this and many other cell lines from cancers. You can pick any chromosome you like, and going to find differences between individual cells. And this very vast heterogeneity is one major consequence of this ongoing process of chromosomal instability. So another way that's very commonly used to catalogue and observe the consequences of chromosomal instability in terms of genomic alterations in cancer is plots a little bit like this one, as sort of a generic copy number plot. We have the x-axis is the genome in a kind of falsely linear arrangement, since that's not what it really looks like. And then we have regions above the central line in red are regions of amplifications or gains where there's too much genetic material, and the regions below in blue are where there's been comparative losses of genomic material. And it's a fascinating process, but it's also, I think, important to understand the mechanisms driving sin in cancer. And we think that it could be important to understand this right across the lifetime of cancer development. So if we think about initiation, the time when a normal cell becomes transformed into a cancer cell, genomic instability has been associated with this earliest stage, although it's hard to unpick exactly what's going on when and in what order. And if we can understand what might be driving that genomic instability or the processes that regulate it early on in cancer development, we may be able to get clues to the etiology of cancer genomic instability, which might give us a clue to the state of the environment at the time the cell transformed, for example, might give us a clue to cancer predisposition factors, in which case we have the opportunity to intervene and act in a sort of cancer prevention manner. So getting insights into that early time of cancer development is very interesting to us. I'm not going to talk about it today, actually, but we have a couple of projects related to trying to understand the role of chromosomal instability right from the very earliest stages of cancer and pre-cancer. And then, of course, the other important time to think about is the time when we can actually do something about it. So when a patient comes into the clinic and is diagnosed with cancer and biopsies are taken, so on and so forth, and treatment options are discussed, at that point is a very good opportunity for us to understand what's driving the tumour chromosomal instability at that moment in time. And that's because there's this idea that we're able to synergize with chromosomal instability generally, or with specific drivers of chromosomal instability, to know which patients to give which drugs to, essentially. So the paradigm here is the use of PARP inhibitors, for example, seems to be particularly beneficial in homologous recombination deficient tumours, which is another driver of sin. And more recently, people are exploring the concept of sensitizing tumours being sensitized to replication stress response inhibitors, such as ATR and WEE1 inhibitors, for tumours that exhibit a high basal replication stress level. But those are the only 2 real paradigms and examples that I can give you, and that's because discovery of additional potential therapeutic vulnerabilities is quite hindered at the moment. because we have a lack of understanding of cancer driver mechanisms in the first place, and then we also have a lack of understanding of the relationships between specific SIN drivers and specific therapies. So there's so many ways we could drive SIN, and that's one of the major problems that we suffer in the field of trying to pinpoint what are the exact drivers. And lately I've been kind of categorizing how I think about the mechanism driving SIN into 3 categories, just to make it a little bit simpler. So we've got what we consider intrinsic SIN. So this is defects, let's say, in processes are usually responsible for controlling genetic fidelity. So essentially anything involved in precise genome replication, organization, and segregation during cell division processes has the potential to drive chromosomal instability if it's deregulated. So this is things like mitotic defects or DNA replication stress. And we've also got emerging evidence that actually the external environment to a cell, so the tumor microenvironment, for example, these stresses may act to drive chromosomal instability or genetic instability as well within the cancer cells. And although this is far less well understood, I think it's something we should be thinking about, because of course it's often difficult to model this sort of pressure in the dish, but it's something we should be thinking about. And then there's a third category, which is a self-propagating SIN, where if you have one initiating SIN event that creates a structurally unstable chromosome, for example, let's say you create a dicentric chromosome, that's now prone to be going on through iterative cycles of continued chromosomal instability. Even if the first event that happened was just one random stochastic event that's not happening again, just the first initiating event can snowball into further genomic instability. A classic example is breakage fusion bridge cycles, for example. So focusing more on the intrinsic side of things, because that's what we've done most of our work in historically. As I mentioned, there's a myriad of processes involved in keeping chromosomal stability intact during a normal cell cycle, involving hundreds of proteins and many key processes, broadly categorized into mitotic defects, chromosomal condensation defects, replicative stress, or DNA repair, although there are many other categories. And the way that we and many others have tackled this is to start looking in panels of cancer cell line models with functional assays and imaging assays, because that's what's really available to us. So we can have a look at chromosome segregation, we can catalogue the types of errors, we can look at the types of DNA damage foci we see, we can measure replication fork speeds and things like this. So we and others have done lots of this, and it's given us certainly some insights into the likely drivers of chromosomal instability in these models. But we sort of hit a wall over the last few years because the resolution of cell biology assays is limited. You can see the defect in DNA damage repair, for example, but pinpointing the exact problem, the exact kinetics that it's failing to resolve a lesion in, or the exact molecular underlying defect in the pathway, you can't really resolve those using cell biology. We also heavily depend on the availability of appropriate representative cancer cell line models, which is still very difficult. More and more nice panels of curated, more recently derived cancer cell line models are becoming available, which we're taking advantage of, but we're still limited to the fact that these are 2D cell line derived models. And the applicability of the results from those to tumour cells in vivo is still really unknown. And it's very hard to do anything more than just mimic a monoculture in 2D conditions, really. For all these assays, you are able to do things like organoids or some co-cultures, but again, it gets much harder to do all the assays that we currently do. So we've been wondering for a while, how can we improve on our ability to catalogue the driver mechanisms of SIN? And sorry, my laptop slightly obscures the bottom of my slide here. So how can we further define SIN mechanisms operating in tumours, which is where we really want to know about those, because if we want to apply them to actual importance of cancer lifetime, like I said, we need to be able to really catalogue them from appropriate samples. So I mentioned that there's another way to look at the genomic alterations caused by SIN, which is these sort of plots of genome copy number changes. And let me just try to move this out of the way so I can see that better. Okay. Our goal is to actually be able to look a little bit further rather than just saying there's chaos and there's alterations, because actually what we see is patterns of alterations, and those are often patterns just within an individual genome. Certain chromosomes will be upregulated and others will be downregulated. Some of those patterns are across tumor types, for example, but they're very enigmatic, and most people, we really don't understand, I'd say the majority of the patterns of genomic alteration that have been observed across cancer. So we really want to learn new ways to start to interpret genomic alteration patterns in order to specifically detect individual SIN driver mechanisms. And that not only allows us to interpret genomes of cancer patients for underlying information about SIN drivers, but also provides a kind of, in the same step, a clinically applicable biomarker, which you could then use to say they have this SIN driver, we should then give them this treatment. So, of course, we're not the first to be interested in doing this, and we've been absolutely inspired by several people's work, particularly that of, excuse me, Jeff McIntyre, James Brenton, and Florian Markowitz, and Nishant Pillay, who've published some very exciting work in this space, working to derive computational models to unpick genomic alteration patterns from whole genome sequencing, for example. I'll just show a couple of examples of the approaches used. But one major problem with these approaches that we and we've in the field suffer is that the etiology of these cancer copy number alteration patterns is not really known and not tested experimentally. So it's always a little bit difficult to have a ground truth for these patterns. The other problem is that, of course, this is a bulk sequencing, an average profile of millions to billions of individual cells, all of which have the potential to carry a vast cell-to-cell heterogeneity, as I showed you right at the very beginning. So we kind of mush this all down into an average profile, and we lose actually the very heterogeneity that we want to interrogate in order to understand drivers of ongoing SIN. So those are two major problems. So, of course, recently we've been absolutely, I mean, the field is moving so fast. It's such an exciting time. And in the last, within the last decade, it's now a reality to sequence the genomes of individual cells at low coverage, but that's enough to pick up large copy number alterations. I've highlighted here two major methods to do this. One from Floris Foyer's group, which is something we've used for many years in collaboration with Floris's group. And then below we have a very nice direct library preparation method, more recently introduced into the field. I won't go into the details here, but happy to answer later. So this is a very similar, essentially slightly different molecular biology to create these single cell genome profiles. And this is now running and established in our lab as well. So we've been able to do quite a lot of this on our own, which also opens up more possibilities for sequencing more cells. So yeah, and theoretically, we may be able to take single cell genome information from tumor samples, and indeed this is happening. So that's really exciting. It's going to for sure provide another layer and reveal the heterogeneity at cell level that's been missing from bulk whole genome sequencing profiles for many years. But we still worry about something, which is that we may be able to subsample a small number of tumor cells and observe the alterations, but it's still a little bit tricky to be sure that the alterations we see varying between cells were caused by, well, we don't really know when they were created. They could have been created yesterday, they could have been created 5 years ago, but then kept at low clonality throughout the population. So it becomes very difficult to know for sure which alterations were caused by the chromosomal instability drivers we have operating right now. And that's important if you do want to synergize with that for treatment. So I'm going to tell you about a newish study from the lab which addresses this problem. It's a preprint, but actually don't hold your breath for the full paper because we're adding a bunch more stuff at the moment. So this is all based in ovarian cancer, what I'm going to tell you about, but we're currently making this a lot more broad because we found it such an interesting, insightful technique to have a look at genomic alterations caused by ongoing SIN. So the basic premise is that we take a heterogeneous population of cancer cell lines, select individual cells from those populations and grow them in individual wells into new populations, which then grow up for a defined short period of time into little clonal populations. We do this actually as part of the microfluidics device, the Cell M1, which I can highly recommend. So it's actually really good for various things, but it's very gentle on individual cancer cell lines that sometimes don't like to be single cell cloned. So we use that and we get very good viability rates back. And we also now integrate monitoring of the clone growth dynamics, which I won't talk about today, but that's also really interesting and revealing. So we started off really not knowing, and this is one of the results that I always remember the first time we got the data back for, because we really didn't know what we were going to see here. So we knew that the parental population, and this is a heat map here, we'll show a few of these. So what it means is that each row along the side here is a single cell, and then along of course, with the chromosomes along the x-axis as well. And then the color blocks are copy number states. So green is diploid, red is an extra copy, purple is a loss of copy. So this is the parental population, and you can appreciate, first of all, of course, there's lots of non-diploidy. It's a rearranged genomically rearranged cancer cell line, not surprising. And there's also a fair degree of heterogeneity. So you can see alterations present in single cells or subsets of cells that vary quite a lot between individual cells. So we really didn't know what to expect when we sequenced the final populations. We estimate they've undergone around 20 to 22 cell divisions by the time we sampled them in this particular case. So I was really thrilled to see, by eye at least, a very matched level of heterogeneity between the cells, which, now we think about it, is not that surprising. We know they're chromosomally unstable. We could see that they were missegregating chromosomes routinely. But of course, the advantage of this copy number profiling technique is we can actually analyze alterations right down to 1 megabase or smaller, which you couldn't detect microscopically, and they may not even be going through a missegregation phase. So we can really get a good deep insight into the ongoing copy number alterations caused by SYN over a defined period. So by all the metrics of heterogeneity that we have been exploring, and I'm sure there's a few more we can do, and I won't go into the details too much, again, happy to take questions, but we essentially see across this cell line and another cell line that we did this in as well, that we quickly re-establish parental levels of heterogeneity just after 22 or so genome doublings. So in contrast to that lovely heterogeneous copy number alteration plot, when we made what we call pseudo bulk, so we take all of the reads from the single cells, mash them up together, and treat it as a single sample to mirror what we would have got if we just sampled that population with an average sequencing method. What we can see is that across the parent and both of the clones, they're essentially identical. I've picked out the only real change that we noted, which is that in one of the clones, this gain on 11q is no longer present at a bulk level. But overall, what you might be tempted to think if you had only done this analysis is that there's no obvious change in the genomic landscape of clones derived from parental cell lines, cells. So, which means we've completely lost all that lovely heterogeneity that we can make use of. So in fact, how can we make use of that? So this is quite a mess, right? So they're very rearranged cancer genomes, and it can be a little bit tricky to pick out what's new on the background of such a heterogeneous, genomically rearranged. So to do that, we just essentially, again, going over this a little bit fast, this is all in the preprint as well. We recalled all of the copy number alterations of each cell relative to what we calculated is the most likely ancestral starting position, because it was the most frequent. And then we can actually pick out all these alterations here. So grey is nothing really changed during the time, and then blue is losses, and red is gains again. And at the bottom here, you can see we have the a commonly used metric, fraction of genome altered, for the two clones, which again is high, but it's also high in the parents. That doesn't really tell us very much. But what we can do now is calculate a much more interesting and useful metric, in my opinion, which is the fraction of genome altered since the clone outgrowth. So that's the exploration space each cell has undergone during those 22 divisions. And then what you can see on the graph here is that, quite fascinatingly, we've got a spread from about 20% up to almost 50 or 60% of the genome can be explored, which means up to 50% of the genome can be affected by a genomic gain or a genomic loss. In such a short time, an individual cell can explore a huge repertoire of genomic alterations. And I think that's really important to think about, that during all this time these chromosomally unstable cells are growing and dividing, they're exploring a huge space. And then we went on to do sort of various analysis of the types of alterations we see, which sizes are they losses or gains. We actually tend to see a lot of everything. And we could look into that more, and we are looking into that more. But we also really wanted to see if we could use this data to ask us a long-standing question that we and others had, which is that can we see evidence of selection for or against certain types or sizes of copy number alterations? Because there's this paradox in cancer aneuploidy, which is that large chromosomal alterations tend to be very detrimental to cell fitness in normal cell systems, but they're obviously providing some kind of benefit in the cancer setting because they're so prevalent. I mean, it's very difficult to understand whether maybe some alterations in cancer cells are still negative fitness. It's really hard to detect negative fitness with computational approaches generally. So to do this, we've taken a very simplistic approach. We don't have yet all the information to be able to time each of the alterations we see very specifically. We don't really know the exact number of cell cycles or the exact number of cell deaths or anything like that yet. So we just split them into very simply older alterations, which we saw the same alteration more than once, suggesting that it had happened at least earlier in the population growth, and newer ones which happened only once that we could detect. So the newer ones here are highlighted in bold, and the other ones are all grayed out. So you can see we have far fewer of the very, very, very new ones. And again, we had a quick look at the features of those. We didn't see anything particularly in terms of size or direction of alteration. But what we then did is we decided to try to do a proportional analysis to look for the way, whether we see gains or losses or small things or big things, and whether that proportion differed between older alterations and newer alterations. So probably the simplest graph to look at is, and sorry again, I don't have my cursor, but the bottom right graph here, where we've done a sort of comparative fold change plot. And what was very clear, again, this was seen in 2 different cell lines, is that we've got this is comparing, so we're getting increases in chromosome scale loss in the more recent alteration proportions. So this tells us that they're happening because we see them in the recent population, but they're actually depleted from the older alterations category. So that suggests to us that, in fact, large chromosomal alterations occur at an appreciable rate, and that adds up to the fact that we see a high rate of large chromosomal segregation errors when we look at these cell lines by microscopy. But they don't seem to last very long in the population. So suggesting that there is, in fact, a negative fitness for these large deletions, for these new sporadically acquired ones, at least. That's just saying that. Okay, so that's very nice. We can kind of catalogue all these sorts of things: position, size, frequency, direction of alteration of all these alterations, and see, catalogue these sort of numerical and quantitative features. And you can go further, and we're doing this as well, of course, and to look, proximity to known fragility features, or classify all these kind of numerical aspects. But we've done some of this, and nothing really jumped out at us, in fact, which made us very aware of the fact that we really don't know what we're looking for. So we have a few known features of the genome which are fragile, which we can go on. But we also we know so little about the genome, why it has inherent fragility in certain positions or in certain contexts or in certain cell lines, etc., or in certain cancer types, which really highlighted that another major area that the field lacks is a framework for fundamental understanding of genome instability features, which will allow the optimal interpretation of all these sorts of cancer chromosomal alteration changes, not just from our work, but from the field in general, to start to pinpoint what they mean. So in order to address that, I'll go through kind of a few studies, or the concept of what we're doing a lot of now as well, to try to build that framework, or at least contribute towards that framework. So now we switch from analysing chromosomally unstable cancer cell lines, and now we take what we call our blank canvas, and we take non-transformed diploid human cell lines from different tissue specific types. For example, RPE1 cell lines, very, very commonly used in mitosis fields and more increasingly DNA damage fields. and others as well, and even more than these as well. And then the advantage of this, of course, is that if we induce specific SIN drivers, we can see that we go from a diploid genome to something that's not diploid, and then we can immediately pinpoint what that change was, which is less easy to do in the cancer cell lines, as I explained. So with this panel of cell lines, we induce specific SIN types. And again, it's these same 4 categories that I mentioned before: mitosis, chromosome organization, replication stress, and DNA damage repair. And of course, we can go way, way deeper than just that broad category. We can go through and interfere with any one of the subcategory processes within these as well. And then we induce this specific SIN driver for 1 cell cycle approximately only, and then take the readout of the genomic alterations caused by that perturbation using single genome sequencing. And it's very important that we only do this short, acute burst of SIN, because we don't want to be conflating any of our results with selective processes. So we just want what happened straight away. We don't want things to be lost due to negative selection or be positively selected due to positive fitness during cell population outgrowth, for example. I know that other people are doing that kind of thing. I think that's a very complementary and important approach. But just to be clear, what we're doing here is just the acute impact, to keep it as simple as possible. And I'll briefly sort of skim over quite historic work now, which got us set on this pathway, because it was a fun project, really. So we took these diploid RPE1 cells. We induced mitotic defects by an acodazole washout strategy, which has been known for many years to induce lagging chromosomes. Here's a little movie, and just at the point at which we're washing out the nacodazole, it's a reversible microtubule depolymerizing drug. You wash it out, cells which have been stuck in mitosis for a while can start to reassemble their mitotic spindles and carry on through mitosis. And they'll do that with a high rate of chromosome missegregation. I hope you can appreciate that. In a couple of the cells in anaphase, you see a lagging chromosome, and in some cases that will resolve into a separate micronucleus. Okay, so I still show this slide, and it kind of makes me laugh now, really, because it's sort of ancient, archaic data in the days, only about 8 years ago, but where we just simply, this is all we could do. So we used the image stream cytometer to count thousands of cells labeled for specific centromeres using centromere-specific FISH probes, and train the software to count spots and things like that. And it was really pretty hard going. But it did give us some clues that we were certainly seeing some biases towards specific whole chromosome missegregation following nacodazole treatment, which we then verified using some single-cell sequencing in collaboration with Flores Foyer's group. And I always show this picture as well because it again was just such a nice thing to see the first time we saw this right down the microscope. If you use FISH probes to either all centromeres as shown in green here, or specific centromeres, as we see here, we have probes in red to the centromere of chromosome 1. You can start to visualize not just that there are missegregating chromosomes, but which chromosome those are. And this is an extreme example, but we really did see very high rates of chromosome 1 missegregating. It transforms into aneuploidy less frequently, but it's missegregated at a very high rate after this particular treatment. We did this across multiple non-transformed human cell lines. It was always the same. Actually, this unpublished data we have is that it's not always the same bias in cancer cells, actually, although we have yet to really follow that up particularly. And to cut a long story short, and this is all published, of course, the reason that chromosome 1 and to a lesser degree chromosome 2 was so susceptible to this nacodazole release treatment is that during the 8 hours that we used of nacodazole treatment, the cells are arrested in mitosis and they start to undergo a process called cohesion fatigue, which is when the chromatid, replicated sister chromatids, don't exactly, well, at extreme cases, they fall apart. And in middling cases, they don't look much different, actually, but they're very prone, for reasons which are not particularly clear, to increased frequency of missegregation once the cell can then go through anaphase. So, and we've looked at other types of mitotic defect as well at the kind of level of whole chromosomes again. And of course, many other people have studied a really nice array of different techniques and model systems to show there are chromosome biases under specific drivers of SIN. And then, of course, we got slightly more interested in modeling more complicated or more perhaps physiologically relevant drivers of chromosomal instability that were relevant to cancer. And from other work in the field, we'd been interested for a long time in DNA replication stress. So just briefly to introduce this term, it's an umbrella term. It's not particularly intuitive what it means, and it's a very broad term as well. So essentially, anything that can impair DNA replication. Some people also like to have the definition that it causes DNA damage. So there's a broad, unclear definition of replication stress, but it seems to be an important driver of sin, or it's at least present as well in several cancer types of interest to us and additional ones these days as well. So particularly colorectal cancer and ovarian cancer is where we first came across it. So you can do the same kind of approach. You can take diploid cells, you can add on a little bit of replication stress by various means. We've used pharmacological inhibitors here. We've used afidacolin at a low dose, so cells will still cycle through, but with impaired DNA replication. And we've also done a short block and release of hydroxyurea, which blocks cells in S phase due to a lack of nucleosides, and then they can proceed through the cell cycle again after release. So we induce DNA damage, segregation errors, micronuclei, etc. And then, of course, we track the changes by single genome sequencing. So we did this with quite a lot of cells, around 350 cells for afidacolin at least. And then what I'm showing you here is what we call a pile-up. It's just a collection of all the alterations that we saw across any of the cells from that population treated with afidacolin. And we just align these along the genome just to see what we can see. So we started to see very gratifyingly some really interesting patterns. So for example, some hot spots where we see consistently the same types of alterations and different kinds of alterations. And also that was different between afidacolin treated cells or hydroxyurea treated cells. And different again if we depleted MUS81, which is another key internuclease important for the resolution of replication intermediates. And it's contrasted with what we saw when we impaired mitosis, where we see much more whole chromosome gains and losses, as I showed you before. So we spent a long time and we're still thinking, why these regions and why the different genetic outcomes. So why some regions always convert to a large chromosomal loss, why some regions translate into a small chromosomal amplification, for example. And to address this further, one of the approaches that we can take is we can think about, well, is this a sequence dependency or a cell type specific feature? So there are many things suggested to be contributing to positions and rates of replication stress, for example, some listed here. So to see if we were looking with on a cell type dependent factor, we did the same thing in another cell line, another non-transformed human cell line. That's just to show you we get the same type of induction of replication stress at a cell biological level. And then this is a pile-up of the two data sets, only showing large chromosomal alterations, which is what we focused on for this study, which is again published. I'll probably put it somewhere. And what we particularly noticed is, although the frequency of large alterations per cell was very similar, the distribution of these across the genome was quite markedly different, with the RPE1 cells having these very specific 3 regions of high fragility, and the BJ cells having much more distributed fragility across the genome. We catalogue various features of the cells, including DNA replication timing, gene expression, etc. And again, to cut a bit of a long story short, what we ended up doing is kind of cataloguing everything we could about the genome and the cell type specific omics that we could get, and lined those up with the positions of the frequent breaks to see if we could see any clues. Why did we get this recurrent break on chromosome 7, for example? So zooming in just one step further again to that very, very specific reason, this is where I really need the cursor, but at the top here you can see these lines with RPE and BJ and with all these triangles. The triangles indicate positions of large alteration, large deletions or gains. And you can see there's lots concentrated there in that left position on the RPEs and not really in the BJ cells at all. The replication timing is below that, and it's actually the same pretty much between both cell types. So it just showed one. It's very late, that whole region. Importantly, just late replication timing per se isn't a problem because the BJ cells don't have fragility in that region, and there are many other regions of the genome that are late replicated that aren't subject to these recurrent deletions. So the key thing that was different in this case was that the expression of a very large gene, ORTS2, is expressed in RPE cells and not in BJ cells. And we found various other examples where the expression of a giant gene over 600 kilobases is our definition of that, and these are often over 1 megabase. That's really critical in collaboration with late replication timing to produce a very fragile site. So none of that is particularly novel or exciting because there's a long-standing known association between large genes and fragile sites induced by phytocolin. But here, the resolution of our approach and the genome-wide nature of our approach means that we can pinpoint much more with much more spatial resolution where these breakpoints are occurring. And it also allows us to look across the whole genome, all of the areas of late replication timing or large gene expression, rather than only being able to focus on cytogenetically visible or trackable sites. So to conclude this section, really just to sort of tie it all up neatly, we wondered then whether this highly susceptible region in RPE1 cells, and the reason it was deletions, could be because that region was highly prone to breakage, and which would cause chromosome segregation, incorporation into a micronucleus, and then be able to potentially be lost from the sequencing, downstream sequencing. And we'd always wonder, because we'd often induced replication stress in cell lines and see these segregation errors, and you always wonder, well, what actually caused that and which chromosome is it? So indeed, when we look at the identity of chromosomes in micronuclei using chromosome fish paints, we see that chromosome 7 is very, very highly enriched in phytocolin-treated RPE1 cells, whereas hydroxyurea doesn't cause any obvious bias in chromosome identity in micronuclei. I mean, this is, again, not that surprising because it all matches up with what we saw from the sequencing, but it really highlighted to us how when you've got a bias in fragility sites, that can really lead to a very highly biased chromosomal evolution pattern, for example. So here, almost half of all micronuclei contain that region of chromosome 7 under this condition. And if you could imagine a similar kind of scenario in a cancer cell line, if it also has a bias towards a certain region of the genome being recurrently subject to alterations, then maybe that's something important to take into account when we interpret cancer genome alteration patterns as well. So we're basically doing a lot more of this with the hopes that when we can start to see patterns that are going to be helpful for us for inferring cancer-specific patterns. So an inkling that this might be feasible comes from the fact that on the left here, what we do is we take information from modeling replication stress in RPE cells, which I just talked through. What we see is this red boxed region of copy numbers points, which are all enriched. These are all large deletions, and they're all very enriched in late replication timing. For example, that's a feature of a type of copy number alteration. And from separate clone-seq data of yet another cancer cell line, we also see a subset of alterations, large losses again, which are highly enriched in late replication timing, suggesting that at least this subset of copy number alterations in this cell line, and these are the most recent ones I should point out as well, are most likely driven by replication stress. And actually that's backed up by cell biology that we have for this as well. So we're doing this now in a much larger scale across many other drivers and different cancer cell types. I think I will probably stop here because I have a bunch more stuff. So I'll do 2 more minutes and I won't do the rest of it. So just to say, this was all focusing on intrinsic drivers of sin, but there's also the self-propagating category. And I just wanted to show you something else that we saw in the clone-seq that was really interesting. So this is back to the first major thing I was talking about, tracking the evolution of the genomes of cancer cells over time by taking single cells and growing them into clones. So when we looked at the most recently acquired copy number alteration pileups, we noticed these kind of regions of recurrent alterations on chromosome 3 here in one of these cell lines. So we wondered, is that because this is a recurrently fragile site, and can we test that with fish? So what we did is we also saw this in the parental cell line actually. So we took the parental cell line, and tried to test this, as I've just said. And again, looking with chromosome-specific paints. And again, so very gratifyingly, we're able to go from the genomics here and validate that with cell biology to show that, in fact, if you analyze the rates of these chromosomes, chromosome 3, 6, and 12, present in missegregating material, that indeed chromosome 3 is very highly represented in cells that make a mistake during cell division. So, and I'll skip through the last bits here. Happy to take questions, but I want to make sure I've got time to hear your comments and questions about the talk. So, in summary, and I apologize for the text-heavy slide, but we think at least that understanding the driver mechanisms of sin in cancer is important for both early stage and late stage cancer prevention and treatment. Cell biology assays are very, very useful, but laborious and limited. Single cell genomics is really exciting new technology to give us a new layer of cell heterogeneity, and to really get the most out of this new information, and the information that will come out of this at a rapid rate, I'm sure, is going to require the understanding of what copy number alterations are actually caused by ongoing sin. So, for example, using things like clone-seq in parallel to sequencing solid tumor samples, for example. And also skipping down really to the last point, the most important point, which is that interpreting all these copy number alterations is going to require significant expansion of our fundamental knowledge regarding the positions at where the genome is fragile and why it is and in which contexts. And here is just to thank my wonderful lab and collaborators and funders. And just before I stop to take questions, I'm going to plug the wonderful next EMBO workshop on chromosome segregation and aneuploidy, squeezed together into chromoploidy 2025 in this absolutely beautiful location in northern Italy, April 27th, 30th, 2025. The website's open, registration is open. Please do join us there. And I think I'll stop sharing, and then I hopefully can see some of your faces and answer some questions. Thank you very much for listening.
Perfect. Thank you, Sarah, for a great talk. We've got a couple of questions in the comments. Please add, please send your questions through or raise your hand so I can find your comments. We've got a question about distinguishing subclonal and unique events. So how did you do this in clone-seq?
So it was very simple. In fact, if we saw it only once in the population, so let's say we'd sequenced 50 cells, if we saw it once in that 50 cell population, we counted it as unique and therefore more recent. If it was present twice or more, we counted it as subclonal and therefore less unique, less recent. There's certainly limitations to that approach, but that was the simplistic approach that we took.
All right, I just have a follow-up question with this one. So how do you distinguish subclone and unique events? So if you only have, think one cell has this event, then you classify it as unique. It can be caused by sequencing error or noise of the data, right? I just don't know how reliable it is.
Yeah, I mean, there's always the chance of a technical artifact. I get that. I mean, I think our controls are cells that we haven't treated with something, for example, in the terms of the technique. Obviously, the cancer cell line, you get this cell heterogeneity, but normally we take diploid cells, for example, and there's not really background noise. So I think it's very rare to get a spurious alteration. There certainly could be some. So that's a concern, but I'm not that concerned about it given what our controls show.
Thank you.
Thank you.
We've got a hand raised by Jeff.
Hi, Jeff.
Hi, Sarah. Great talk.
Thank you.
I wanted to get your feelings on how quickly you think selection is acting on these events in vitro.
Well, as I said, I mean, we see our data seems to support the notion that large chromosomal alterations get notably depleted from the population after 22 cell divisions. That's a period of a couple of weeks. So perhaps much faster than I would have thought. I mean, many factors are going to govern this, I suppose, like how detrimental are they? Are we talking the cell dies straight away? Or if it's just a slight reduction in the proliferation rate of that lineage, you might expect that to take longer. So we might, if we could time that a little better, to be able to say not just it isn't there in the kind of vaguely earlier population, which is something we're working on, then we might be able to get a better time frame over which these selection events are occurring, and then try to understand the mechanism. And we're
Yeah.
also tracking now, because I said we're kind of I didn't show you data, but we're tracking the clone growth over time. We're also tracking cell death markers over time as well. So we'll hopefully have that information alongside these data as well.
And do you think that, just sorry, quick follow-up question. Do you think the same behaviors would be observed in the earlier phases of chromosomal instability in terms of tumor evolution? Do you think working with these cell lines have a kind of genome structure that they favor, they want to return to, whereas perhaps earlier on in evolution you have less selection acting? Or
yeah, I mean, I guess the way to do that again is sort of doing this is to look at other perturbations or early. So we've got some model systems of early, what we consider to be early cancer development, and there it will be very interesting to see when they're sort of not even cancer yet. How does the selective pressure act on these alterations? Yeah, I think I get what you're saying anyway. Yeah, I don't know. Yeah, I think we've got David. Hi, David.
Oh, great. Hey, how's it going, Sarah?
Good. Nice to see you.
Yeah, so I wonder if you could comment on the frequency of whole chromosome missegregation that's occurring spontaneously, as well as also the widely cited propensity or not of centromeric breaks to occur.
Mm-hmm. Great question. So whole chromosome alteration rates, we can measure that by obviously microscopy, and we can look for lagging chromosomes with centromeres. That's sort of the best marker, which generally across all cancer lines that we've analyzed, at least under our imaging conditions, is really low. It's much more frequently acentric or bridges. Now in the clone-seq, it's actually a little bit tricky to pull those out, I'd say. One of the criteria we use to deciding if an alteration is new or not and non-clonal relative to the chaos there is we look for a unique breakpoint. That's one of the methods we use to be sure we're not just calling an alteration which is just because of a miscalled alteration in copy number, and also because we have this major problem. So we're undercalling in that sense. And then the other problem is, of course, that most chromosomes in cancer are no longer just the chromosome they used to be. They're often a fragment of that joined to another fragment, often multiple chromosomes, components to a single, cancer chromosome. So that means that, of course, if that whole cancer chromosome gains a whole copy, we won't see that like a whole unit changing. We're going to see several pieces changing in concert. Now, with the number of alleles there are, the number of differences between cells that there are, and the low sampling frequency we have, we won't see those patterns often enough, because I often think about this to say, well, we always see those co-occurring. That's probably a deranged chromosome, and we can understand that that probably means a whole chromosome gain or loss. Anyway, long-winded way of saying we can't really be sure if we're detecting it properly or not. But from the cell biology, it seems to be a lowish rate. I mean, not always, though. We've got some cancer cell lines or some clones of cancer cell lines that's not quite true for.
Great. And how about the centromeres?
Yeah, centromere breaks. Lovely question. So we haven't seen that particularly in the data we have so far, but I think it would be very interesting. I mean, of course, that is the most common place for there to be an alteration in copy number in chromosomes in cancer genomes. So whether it happens early on, though, and then it's kind of, then they've done their thing, they're now translocated to a different chromosome or whatever it is that's explaining that, it doesn't happen again, or maybe it does. I don't know. We didn't pick it up in the most recent data set, but we'll have another look back at that. Thanks, David.
Okay.
We've had a question come through asking about how fragile regions are defined.
Yeah, good question. So, I mean, there's various fragile sites defined. There's the common ones, which is the common fragile site. So they were historically defined as regions of metaphase chromosomes that appear as gaps or breaks when you make a metaphase chromosome spread after the treatment of very commonly afidicolin, but sometimes other perturbations of replication. And they're so-called common because you see them frequently enough to identify them, and they're frequently seen between donors for humans, essentially. And this was nearly all done in lymphocytes. And so there's some very, very common fragile sites which seem to be kind of independent of tissue type or person to some degree. There's also early replicating fragile sites, which have been defined slightly differently, and they tend to be early replicating, whereas the common fragile sites tend to be late replicating. And then, of course, the cell type specific fragile sites, which are cell type specific as well. So again, historically, these have been catalogued using kind of laborious cytogenetics, and it's sort of still not fully catalogued, I'd say. And I think in a way our afidiocolon followed by single cell genome sequencing quickly pulls out cell type specific landscape of fragile sites to some degree. It's only the ones that break and causes a copy number, of course, copy number alteration.
I have a question as well. With your data, where it was really nice where you're showing your combined data of the single cell sequencing and the actual images of the fish labeling of your chromosome 7 migrating to the micronuclei, and that being linked to a losses of chromosome 7 in the sequencing data. Do you think that because you're seeing both of those, the sequencing isn't capturing the micronuclei and you're only capturing the main nuclei?
Yeah, I mean, in that particular sequencing data set, yes, we think that's the most likely explanation. I think in that particular prep, they did nuclear prep, so most likely do lose the micronucleus from the cytoplasm, unless, of course, the micronucleus sort of sometimes sticks to the nucleus. So not 100% sure why that's happening. I have to say, though, I think we see a similar bias towards loss rather than gain in our sequencing as well, and we don't in most cases do nuclear prep. So I still have a sneaking suspicion that it could be something more than that. It could be that for some reason, at least during we use a tagmatase based method, it could be that there is actually something refractory to sequencing in the micronuclei. We haven't tested that specifically yet, but in general, if we see something going in the micronucleus, we tend to see losses rather than gains. It could be that it's not getting sequenced, or it could reflect an ongoing process which that material is much more likely to get lost from the population because it'll get degraded or lost or whatnot. So I think it's probably multiple factors explaining why we tend to see losses rather than gains.
No, it was a really clear talk. I think the other questions have mostly been answered by others.
Can I ask a question? I don't know if that was answered or not. So I'm interested in knowing why certain CNEs co-occur, and if there is a sequence, they always follow, or they can jumble up even when they co-occur.
Yeah, it's a great question. I mean, I think there's been some really wonderful studies looking at the kind of deterministic evolution of copy number alterations over time. So taking diploid models and inducing SIN and tracking the kind of collection of accumulation of copy number alterations over time that's been done in gastric organoids with P53 loss. It's been done in breast and kidney cancer cells by Steve Elledge. So it seems that there's some kind of pattern to which copy number alterations tend to be maintained. And obviously we've shown that there's a pattern to some chromosomes being gained or lost at a time. In terms of ones that co-occur, I think that's not something we've seen in terms of our kind of acute induction. And as I was saying to David, we might, if I feel we lack the resolution to be able to say certain copy number alterations co-occur in the cancer cell line experiments we've been doing because of the resolution and the depth we've been probing the populations with, because there's just so much heterogeneity. So, you know, if you think about those fish plots at the beginning as well, you know, every allele of every chromosome in every cell could be quite different. So, you know, seeing what is happening in ongoing SIN is really, really complex. But I think measuring the patterns of alteration that seem to like to happen over time or in combination because of the sort of selective pressure is a bit of an easier question that's definitely seems to show some interesting patterns there. I hope that answered your question. It was a bit vague, sorry.
Maybe I can ask a quick follow-up.
Yeah.
So you also sort of said replication timing is determining. Is there also heterogeneity associated across different cells for replication timing, or is it the same? I mean, we expect heterogeneity in every aspect then.
Yeah, so it's sort of largely the same and a bit different. And that actually seems to hold true for normal cells and cancer cells. So let's say 70 to 80% of the genome is pretty much the same. And then some regions will be different, some might be earlier or later. And there's a couple of nice studies come out recently, actually cataloguing replication timing across different cancer cell types. So there are differences. It's still mostly the same, which I do find quite interesting, actually, given the absolute chaotic rearrangement of different cancer cell lines to each other and to normal cells.
Thanks.
Thanks.
I guess we can have this final question from
Yeah.
Mohammed, and then
Yeah, thanks so much for the presentation. I have a very basic question. Now I'm studying breast cancer initiation in vitro, and we have a primary cell. And we try to treat them with different hormone and chemicals to study the cancer initiation. But we
Right.
look at cell population because we have a different epithelial cell subtype. After this presentation, I'm thinking about using a chromosomal instability or copy number variation. Can we do it? And do you think passaging affected that process as well? Because in primary cell, we just can do 7 to 10 passages.
Yeah. I think you can definitely see things there. I think that sounds like a really interesting model. It's great that using primary cells. Yeah, we definitely, I mean, I would recommend doing clone-seq or something similar, or even just a short period of exposure to your driver or your hormone treatments, and then just analyzing a subset of the population. Or you could do some microscopy as well. I mean, you can please feel free to drop me an email and I can chat to you more about this. But I think that'd be super interesting to capture those very, very early cell divisions to see if you're getting any instability from those hormones. I think that'd be really interesting.
Yeah, thank you so much. Yeah.
Thanks.
perfect. So I'd like to say a big thanks to Sarah for giving a really great talk today, and so thank everyone for attending and some really good questions.
And thanks so much for Amy, who's done an absolutely wonderful job chairing. This might be your new job, Amy. Good job.
Perfect. Okay.
Hi, Steve. How you doing?
Very good. Super talk, Sarah. See you soon, I hope. Thanks,
Yeah,
Amy.
bye-bye.
Thanks, everyone.
Bye-bye, everyone. Thanks a lot.